Each muscle cell is made up of multiple bundles called muscle fibers.
In this model, you can see the sarcoplasmic reticulum covering each fiber and transverse tubules or T-tubes interwoven between the fibers.
If we look at just one fiber, we can examine the process by which it contracts and expands.
Inside the fiber is actin and myosin, two proteins that will pull the fiber together when calcium is present.
If we zoom in even further, we can see how calcium release and reuptake is regulated. On the top left is the T-tube, on the top right is the sarcoplasmic reticulum, where calcium is stored and bound by calsequestrin. Each calsequestrin holds up to 50 calcium molecules. On the bottom is actin and myosin. The process begins when the T-tube is depolarized by a stimulatory neuron. This induces a change in the conformation of the dihydropyridine receptor, or DHP, which is located on the T-tube membrane. DHP is coupled to the ryanodine receptor, which is located on the sarcoplasmic reticulum membrane. When DHP changes conformation, it causes ryanodine to change conformation as well, allowing for the efflux of calcium. Calcium then binds to troponin, which frees up binding sites on the actin for the myosin heads. These myosin heads pull actin molecules together, constricting the cell. Calcium is constantly binding to and disassociating from troponin. When the electrical signal from the T-tube subsides, ryanodine closes and Serica takes control. Serica is an active transporter that constantly pushes calcium, when present, into the sarcoplasmic reticulum. Calcium storage is optimized by calsequestrin, which binds calcium and increases the concentration gradient. When multiple muscle fibers are stimulated simultaneously, the entire muscle constricts. Each time a hummingbird flaps its wings, a wave of calcium rushes in and out of the sarcoplasmic reticulum. Considering how fast a hummingbird flies, this process is truly remarkable.